Good morning, everyone. I'm Zena Wolf. I'm a PhD student here at UC Davis, and I'm in Dr. Danica Banish's lab, who Dr. Dickinson spoke about finding the Breakey mutation. And today I'm going to talk about the work that I do in cleft palate in dogs. So cleft palate, it's a developmental defect that results in the failure of the roof of the mouth to properly form. Often with cleft palate, you'll also see a cleft lip, which is a failure of the lip to properly form. These are both developmental defects, and you can see them pictured here. So here you're looking at the roof of the mouth of a dog, and on the midline of the palate, you can see that the mouth just doesn't come together properly, and this is a cleft palate, okay? And if the lip doesn't come together properly, you'll have a cleft lip. Okay, and this is also pictured here in this bulldog puppy where you have a cleft lip on both sides. This is something that you might notice right at birth, especially if the dog has a cleft lip. It's very visible. You can see that in the bulldog here. But if you have something such as a cleft palate, it might take a little longer to notice. So you might notice that the puppy has difficulty nursing, or milk will start to bubble out of the puppy's nose, and that'll kind of key you in to open that puppy's mouth and look at the palate. Other things you might notice are something such as a runny nose or nasal discharge, which you see in this little pup down here. Some dogs will also start to cough a lot, and they can develop aspiration pneumonia. So cleft palate results in communication between the mouth and the nose. So food will get into the cleft, get into the nasal passages and into the lungs, causing infection and aspiration pneumonia. You also might notice that the pup just can't keep up with gaining weight like the other litter mates are. This is because they're just unable to nurse and they can't put on the weight. There are many causes of cleft palate. There are environmental causes, just to name a few. There are medications that a dog might take during pregnancy or exposure to pesticides. And today I'm going to talk about some of the genetic causes that we found in our research. So there are certain breeds that have a very high prevalence of cleft palate. This includes all brachycephalic breeds. We also see a handful of other breeds that are listed here. We see a lot of Whippets, Labs, and what I'm going to spend some time talking about today is the Nova Scotia Duck-Toy Retriever. That's where we've started our cleft research. But why is it common in certain breeds? And really the way to answer this is, it goes back to why we love our breeds so much, because we've designed them the way we want them. And it goes back to breed creation. And we can see that there's a lot of large diversity among breeds that we have done by selecting these dogs over time. And there are both good and bad consequences to this. So many of you know how breeds are created, but I'm just going to kind of go over it. So breed creation was a result of founder effects. So it means that you take a very limited number of dogs to start a breed, and that is the entire gene pool that makes up the breed. So if you use an example such as red and yellow ladybugs, you take this population and you just select a handful to create a new population with. If you pick the red ladybugs, you're only going to end up with red ladybugs. So you're losing the variation that might be in those yellow ladybugs. So this is definitely good for certain traits, but also can cause a high frequency of disease. Dogs have also undergone population bottlenecks. This further limits the genes that you might see. So there are a couple very popular or very well-known population bottlenecks that dogs went through. One was during the World Wars. So at that time, people couldn't really afford to keep dogs, so a lot of breeds started to kind of die off. And when they did want to go back and repopulate the breeds, there was a much smaller gene pool to start with. And you also have the effect of the popular sire. So if there's a very popular sire, this might be, or just a handful of sires, they're bred to a majority of bitches within that breed, and you're going to end up with just that gene pool. Again, so this can be seen if you take a bottle full of marbles, lots of genetic variation, and you pour out a few marbles and they get stuck in this bottleneck, you end up with just a handful to kind of populate with, and the resulting population is not what the original population was. So as a geneticist, this is actually a very good thing. We can really take advantage of this and try and find genes that cause mutation and cause defects. So The laboratory that I work in, we started our cleft palate research in the Nova Scotia Duck-Tolling Retriever. It's a very small breed of dog. And in 2002, a health survey reported that 7.2% of tollers produced litters with cleft palate. So it's a very high frequency. We wanted to investigate this. Now, just a little bit of a background on this breed. So they underwent a founder effect. They had a few number of sires that started the breed. And they also had a population bottleneck. So in the 1800s, disease and kennel fires almost wiped the breed out. So that was what they started with. So we wanted to study cleft palate. And today I'm going to talk about what would happen if you guys sent me a sample and the steps that I would go through to study cleft palate and how we did this in the toller. So what you need to do, first of all, we're going to gather information. We want to know about what the cleft looks like. And then we're going to go in and use genetic tools to study DNA, which I'm going to tell you about today. And then we want to look at the inheritance and the frequency of what we find. So we want to know how cleft is passed from generation to generation, and if we can create a genetic test to help you guys breed against this. So the first way we gather information is we want to look at the physical characteristics of the trait. Okay, this is known as the phenotype. And in the toller, we were seeing, of course, cleft palate. So pictured on the top, you have a pup that has a cleft palate. It's a wide opening in the mouth when compared to its normal litter mate. We also go in and we do a little more advanced imaging. These are micro CTs. So you're looking at the roof of the mouth of a puppy. These are 3D reconstructions of the bones, and the blue is the palate, okay? And you can see that the palates just don't come together the way they should when compared to the bottom. Micro CT also allowed us to notice that these dogs had short and lower jaws, so there were additional abnormalities. So on the top, this pup has an overbite when compared to its normal litter mate. Some tollers also had a cleft lip. So here you're looking at the nose of a pup, and this is similar to the first dog that we saw that had a cleft lip on both sides. Often dogs with clefts also have additional birth defects, so we always want to investigate those. And we noticed that a lot of our tollers have fused toes. So you're looking at a normal toller, 4 toes, and when compared to the affected puppies, you can see that they look like they only have 3. We do know that the bones are there. We did do x-rays. Everything is there that should be, but they're just fused in the middle. So before I go any further, I just want to make sure we're all on the same page, and I'm going to go over genetics for a little bit. So if you're a human or a dog or a plant, we are all made up of cells, and these cells contain nuclei that contain all of our genetic information. The genetic information is organized into chromosomes. Dogs have 39 pairs. That's what you're looking at here. And within these chromosomes are the genes. And genes are encoded by base pairs. So these are the A's, G's, and T's and C's, okay? Every dog has 2 copies of every gene or set of A, G, T, and C. So it's these base pairs, this DNA sequence, that we're interested in finding mutations in. And all a mutation is it's a change in a base pair. And that's what we want to identify. And this can downstream lead to the various defects that we're seeing. So we use certain tools to find these changes. One tool that we have is the genome sequence. So in 2005, Tasha the boxer was sequenced. Okay, it was found that she had 2.4 billion base pairs, and they encoded for approximately 19,000 genes. So without this roadmap, per se, we would have no idea of what we were looking at. So we really need this tool to go in and know what genes we're looking for and understand what mutations that we possibly can find. And we also have genetic markers. So a genetic marker is a base pair sequence, or an AGTC, that we know the location of. So we know exactly where this marker is with respect to those 39 pairs of chromosomes. And they can be different between any 2 individuals. They're not necessarily always disease-causing, but they can help us identify disease. So if you take this picture, for example, you have a base pair sequence, and you have a healthy woman that has a G, and then the sick woman has a C. So there might be some reason as to why she has the C that's causing her disease, and that's what we want to look for. So we can use these to identify regions that we want to investigate. So this is a Manhattan plot, as Dr. Dickinson was talking about before. You know, a perfect Manhattan plot looks like the New York skyline, but mine, fortunately enough, looks a little more like a Dubai plot. So I'm very grateful for that. But so what you're looking at here is that you have the genetic markers per chromosome on the x-axis, and then we run statistical associations to see if we can identify a chromosome that might be of more interest than the others. And fortunately, we were able to do that. And it only took us 22 cases and 107 controls. So that's a very small sample set compared to something such as a human study. So here we have a region on chromosome 14. We also have a smaller association on chromosome 27. So because we have this plot, we now know that we can look at chromosome 14. We know exactly what this region is. We can go in and we can pick out genes that might be of interest, that might cause cleft palate. We can also do the same for chromosome 27. So because we know the region of interest, we then go in and we look at the genes that are on the chromosome. And Tasha has provided us with this. So we know what's in the region, and then we also use knowledge from additional experiments that have been done in other models about the genes. So often a lot of work has been done in the mouse model, and fortunately for us, within one of those peaks, we had a gene in the region that causes cleft palate in a mouse. So we started looking there, we sequenced all the base pairs, and we found a mutation. So this mutation causes cleft palate in mice, and it also causes cleft palate in dogs. This isn't always the case. Sometimes there is not a perfect candidate gene. So you do have the option of going in and doing whole genome sequencing. So this is where we go in, we sequence all of the base pairs. So it's 2.4 billion base pairs. Back in 2005, when Tasha was sequenced, this was not something that could be done. It was very labor intensive, very costly. But now it's becoming much cheaper, and we can do it on many more dogs. So if you don't have an obvious candidate, you can go in, you can sequence these dogs, and that's what we were able to do. We found that there was, again, another mutation. So we identified a mutation in a gene that actually is involved in formation of the palate and the fingers. So that explains why these puppies have these fused toes. So we found these two mutations. It's important to understand next how they're inherited and if the mutation is in any other dog breeds, because that's what we're all interested in. So first of all, these dogs had what we noticed were recessive mode of inheritance for both mutations. And that means that both parents are carrier of the mutation. So autosomal recessive just means that you have one good copy of a gene on your chromosome, which is in the blue, one bad copy of the gene, which is the white gene. And that means that both your parents carry it, and they pass on the bad gene to make you affected. Okay, so here you're looking at a Toller pedigree. You have the sire in a square, the dam in a circle, and they're connected. This was a mating, and these are the offspring. The filled in dark black circles are the affected dogs, and you can see there's quite a high frequency within this litter. We also want to know if this is shared with any other breeds. So we have quite a sample set across breeds. We have 30 additional breeds. Here's just a small list. And unfortunately, we weren't able to find the mutation in any other breeds. So it is private to this breed of dog. So we have this knowledge. We know it's not shared in other breeds, but we wanted to know if maybe children had it. So cleft palate is one of the most commonly occurring birth defects in children. It affects 1 in 700 live human births worldwide. So when we looked at the same genes that we found mutation in, we were able to find a mutation in children. And children with this mutation have what's known as Pierre-Robin sequence. And it's characterized by short and lower jaw, which you can see pictured here, and that's identical to what we saw in our puppies. They also have a cleft palate, again, identical to what we saw in our puppies. These children also have a tongue that falls back into the throat. We unfortunately weren't able to observe that on the puppies, but we don't know for sure. So what's the result of all this? So with the Tollers, we now have 2 DNA tests. They're offered through the Orthopedic Foundation for Animals. So if you want to breed a Toller, you can submit a DNA sample, they will test the dog for cleft, and you can make an informed breeding decision based on these tests. We were also able to identify the genes of interest for cleft palate in children with Pierre Robin sequence. And our next step is that we really want to do this for other breeds. We want to look in bulldogs. We want to look in all the brachycephalic breeds. We know they have a high prevalence, and that's where we're going next.
How much does a test like that run?
The Toller cleft palate test is about $70.
Oh,
And that's for both mutations.
okay. And that's per dog?
Per dog, correct.
Do you recommend both the DNA and the sire?
I do, yes. Yes. You know, we're encouraging that you don't breed two carriers of cleft palate. So what's next? We really need samples. We can't do this without you guys. We have started to collect samples. There was an article appearing in a newsletter in June that reached out to the brachycephalic community. Maybe some of you saw it, that we just need samples to get this going. And we have talked to the Bulldog Club of America. They are willing to help pay for sample submission. And if you don't have an affected dog, the control dogs, the healthy dogs are just as important. We cannot find the mutation without something to compare it to. So if you're interested in submitting samples, please contact me. My email is up here. Or if you have any questions, I'm more than willing to talk to you about this. So with that, I just need to thank Dr. Danica Banish. She is my PI and the one that allows me to do all this research. And of course, all the collaborators, because this cannot be done alone.
Your email again?
Oh, yeah, of course. So if you have an affected dog, we will take the puppy, but we also would need blood. Unfortunately, swabs just aren't good enough to do the testing that we need to do.
So you need like serum or like?
We actually need unclotted blood. So we need purple top tubes, EDTA.
So would you want us to bring the animals in so that you can draw the blood?
Definitely. You can give me an email. We can set it, or you can send me an email, and we can set up an appointment, and we can talk about that. Or I can come to you if you're local. I do. I have been to a few breeder specialty shows to collect samples. We can definitely set that up. If you know of one coming up, contact me. I will show up. Yeah.
So could somebody come to like their general practitioner and then have it transferred like through IDEX?
We typically don't use IDEX. We'll have it shipped in. But yes, you can go to your vet.
Their GP, then like we could do the blood draw and then we could send
Correct.
it to you guys. So that
Correct.
way they don't, okay.
Correct.
Are you only taking samples from dogs that have an obvious cleft?
No.
See the mutation and where it is?
Yes, yes. So some types of cleft are not as obvious. They can be underneath the tissue. At this point, we're not really able to catch that as much, mostly because you're not seeing it. The puppy isn't as affected as maybe a puppy with a full cleft would be. But that's why we collect the information. We want to know what we're looking at.
It has to be for puppies?
No, not at all. No.
How many mils were you saying?
2 to 3?
2 to 3?
Yeah. Do you have a dog with cleft?
No.
We
She
were
said
like,
she's
yeah.
so good with blood draws. I was like, here, take
Yeah.
samples.
Yes.
Research, take
Yes.
samples. She's awesome.
Any more questions? Okay, thank you.